I'm Sudha Pandey and I'm doing my master's in biomedical informatics at University of Nebraska, Omaha. Today I'm presenting some of my research in the reproducibility of computational analysis to understand microbiome composition and functional profile. Let's begin with what the microbiome actually is. Our bodies host trillions of microbes, including bacteria, viruses, and fungi, living in our skin, in our mouth, and especially in our gut. These microbes aren't just passive passengers. They help us digest food, regulate the immune system, and some studies even suggest they influence our mood. The gut microbiome is so complex and influential that it's often referred to as our second brain. To study this complex community, we have two main approaches. Target sequencing, like 16S RNA gene sequencing, which focuses on bacterial identity, and shortcut metagenomics, which sequences all DNA in a sample given more detail but also more complexity. The first step in either case is a FASTQ file, the raw data straight from the sequencing machine. While microbiome research is exciting, it comes with major challenges. From experimental side, how we collect and handle samples matters a lot. Contamination can easily skip the results, but computation side is just as tricky. Metadata might be incomplete or inconsistent, and there is an overload of tools with poor documentation. And because most tools are open source, reproducibility becomes difficult. That's why this project focuses on implementing a clean reproducible analysis pipeline that can actually be shared and reused. Here's what I set to do in the project. Reviewed literature to understand common pitfalls in microbiome reproducibility, implement a reproducible pipeline for analyzing 16S rRNA data from raw FASTQ files to final insights, and validate this pipeline on a real-world data set from a colorectal cancer study. This hands-on implementation helps bridge the gap between theoretical reproducibility and practical execution. The pipeline I implemented follows a clear multi-step structure. Pre-processing, we clean the data, trim adapters, and check quality. In time to core analysis, it includes denoising, taxonomic classification, and generating diversity metrics. Using sub-analysis, we use LeftSafe for biomarker discovery and PyCrust2 to predict functional profiles. In statistical evaluation, we interpret the output with tools like R or Python to identify significant patterns or association. It's scripted and documented to ensure full reproducibility. Pre-processing, this is first step like preparing ingredients before cooking. We start with raw DNA sequencing reads in FASTQ format, which might contain noise, adapter sequences, or low quality reads. To clean them, we use FASTQ to check quality, multi-QC to summarize the reports, and cut adapt to trim off adapters if there's any. The manifest file acts like a sample for QIIME2. Our goal here is to ensure only high quality, usable sequences to move forward. The output includes clean reads and interactive quality reports in .qzv format for inspection. Here is a screenshot of a multi-QC report from one of my research. Now that the data is clean, we analyze it in QIIME2. First, we denoise it using DADA2, which corrects sequencing errors and outputs a feature table showing how many of each microbe is present in the sample, and representative sequences showing the actual DNA. Then we perform taxonomic classification using Silva database to assign species names and those sequences. This gives us a list of microbes in each sample organized by taxonomic labels like phylum, genus, and species, basically identifying who's at our microbiome party and how many guests are there. In this step, we compare how diverse each sample is internally using alpha diversity and how different samples are from each other, beta diversity. For alpha, we use metrics like standard index, observe features. For beta diversity, we use Bray-Curtis and unifractances visualized in PCOA plots, which are like scatterplot plots that cluster similar microbiome communities together. To make fair comparisons, we normalize the data using rarefaction or SRS. After identifying who's in the data, we ask what they are doing and whether they differ from tumor and normal samples. Let's say helps find biomarkers, microbes that are significantly more abundant in one group like Fusobacterium in tumors, and Picros 2 predicts microbiome functions, telling us if certain microbes are involved in digestion, immunity, or other biological pathways. These tools provide functional insights beyond just presence or absence. To confirm that our findings are not just due to chance, we use statistical tests. Kruskal-Wallis checks if alpha diversity differs between the groups, and Paramanova test if overall microbial composition differs. ANCOM identifies specific microbes that are differently abundant, and these methods gives p-values, helping us know whether the observed differences are statistically significant. Here is the overall end-to-end pipeline overview. As we can see, we have pre-processing, time to denoising, sub-analysis, and statistical verification all in one. Now we put the pipeline to test to finally prove that our pipeline works. We tested it using a real colorectal cancer dataset. We have the bioproject ID and SRS study ID here. The sample had 102 human gut sample, normal versus tumor for colorectal cancer study. We processed the data and compare our results to the published paper, and the same key findings were observed. Similar kinds of microbes were enriched in tumor versus normal, and diversity patterns somehow matched. To evaluate reproducibility, we compared our pipeline with the original study across several components. We used Time2 version 2022.2, which is more modular than older Time1. Instead of the outdated green genes, we use Silver database, which offers better taxonomic resolution. And for statistical analysis, we used R, while the original study used R, while we used Time2's Permanova plugin, which implements the same method. For biomarker discovery and functional prediction, we use the same tools, though ours were new versions, which brings better functional prediction accuracy. In the microbial composition differences, we highlight the overlap and differences in microbial composition between our study and the original. Both pipelines detected major phyla such as Bacteriorota and Proteobacteria. Specific genera like Faecalibacterium and Bacterotoids were found in both studies. However, some microbes were unique to our pipeline, likely due to better detection from Silva and DADA2. A few were only reported in the original, possibly due to differences in database or filtering algorithms. In both pipelines, Fusobacterium and Campylobacter were enriched in tumor samples, while Faecalibacterium and Blotea were more abundant in normal samples. However, our pipeline identified additional taxa group and likely due to improved resolution from newer tools and reference data. This shows that while the core biomarkers were consistent, we were able to gain extra biological insight, demonstrating the power of modern pipelines. This table summarizes how our results compared overall. We used newer tools like Time2 and Picros 2. As a result, we got more detailed taxonomic and functional information. While the match wasn't perfect, for example, a few alpha diversity metrics differed, overall our results were statistically valid and reproducible. That means we could confirm the key findings of original study with the updated tools. To summarize, we implemented a reproducible microbiome analysis pipeline that was validated on public colorectal cancer study. We were also able to identify known biomarkers and discover additional taxa of potential clinical interest. This shows that our pipeline works and can uncover both expected and novel insights. The reproducibility aspect is important here. We matched many findings from the original study despite using different tools. For future improvements, we plan to extend the study in three ways. First, by applying pipeline to more datasets, we can verify if biomarkers are consistent across the population. Second, we want to add machine learning based classification to see if microbiome signatures can predict tumor status. Lastly, we'll use containerization tools like Docker to package our pipeline. This will make it easier for others to run it on their own machine and ensure reproducibility. To finally say, trust your science but verify first. I'd like to thank Dr. Cooper, who is my advisor, for guiding me throughout the project, and my bioinformatics lab at University of Nebraska, Omaha, where I work. And I would like to acknowledge Department of Biomedical Informatics, a School of Interdisciplinary Informatics at UNO, for funding this project. And the whole pipeline is in GitHub link. Thank you so much for listening, everybody. Hope you have a great day.